Hello, I'm Jim Cornell, and this is Base to Base, the weekly biotech podcast from Air Coastal Media. Yes, welcome again to the Base to Base Biotech podcast, and this is number 26, going out on September the 12th. It's Video Games Day, World Dolphin Day, and it is Stand Up to Cancer Day. And to look like we know what we're doing, all of our interviews today relate to companies tackling some aspect of cancer. And it is also a bumper episode with 3 interviews. My voice is still a little bit hoarse, something I don't really need, as I'll be doing quite a lot of interviews next week. Maybe I can get an AI voice to help me out. As there are 3 interviews coming up today, I'll keep the chat to a minimum and keep things moving along. Last week we had just the one conversation because it was quite long, and that was with Microcycle Therapeutics Chief Data and Information Officer John Castle. And as I mentioned this week, we have 3 chats with Poolbeg Pharma, Kling Bio and Curve Therapeutics on various aspects of the fight against cancer. If you do have news or suggestions for who should be on the podcast, or indeed any other suggestions or comments, please do get in touch via jim@deeptechdigest.com, which is also the address you can use if you're interested in advertising with us or sponsoring one or more podcast episodes. Alright, first, as always, let's take a quick look at the news you may have missed over the past week at deeptechdigest.com. Lilly's obesity drug has seen success in its 3rd phase 3 trial. An antibiotic-free bacterial vaginosis treatment has received the CE marking, and we had an article on telomere protection and the fight against cancer. Regeneron is advancing its allergy pipeline to combat cat and birch allergies. Pfizer and BioNTech say there is a robust immune response with their COVID-19 vaccine, and there have been positive results from a trial for chronic gout. Johnson & Johnson's drug could be a turning point in how we treat EGFR-mutated lung cancer. And there was another article about Eli Lilly as it is hoping its new billion-dollar platform leads to new medicines. A clinical trial has provided some positive results for a phenylketonuria treatment, and a novel drug target has come from a Barth syndrome study. Servier has acquired a potential treatment for Fragile X syndrome, and you can read all of these and plenty more at deeptechdigest.com. So let's get down to Stand Up to Cancer. Of course, cancer sadly needs no introduction, and there are many days and months dedicated to awareness and fundraising, as indeed there should be. And of course, we could very easily have featured dozens of companies all trying to beat this awful and ubiquitous disease. So for Stand Up to Cancer Day, first we'll talk to Curve Therapeutics, which is using its gene-encoded Microcycle discovery platform to build a pipeline of cyclic peptide and small molecule drugs to take on cancer, among other things. To tell us more is Simon Kerry, co-founder and CEO of Curve Therapeutics. Hi Simon, thanks for joining us, and I'm looking forward to learning more about Microcycles. But first, maybe could you give us a bit of background on the company?
Yeah, so Curve's a company, it's about 6 years old. It was founded out of research at the University of Southampton by my co-founder, Ali Tavasoli, based on some technology he'd been working on first at Penn State University in the US under Professor Steve Benkovic, and then at that time about 15 years in Southampton. And at the heart of the technology is the ability to screen libraries of compounds, microcycles as we call them. So these are small cyclic peptides, typically 5 to 11 amino acids in size. Most of the development of the system we've done using hexamers, so 6-membered cyclic peptides. And the ability to screen those directly inside cells, so we can screen them against targets in their native conformation inside the cell, so the targets in its dynamic forms, it's correctly glycosylated and other modifications, it's got all its cofactors and everything there. And that's the technology that we thought was a good starting point for a company. And between us, Hallie and myself, we raised a seed round in June 1999 and started to build the company from there, initially in Hallie's lab in Southampton. And now we have a place of our own at Southampton Science Park.
Could you tell me a bit more about your Microcycle discovery platform?
Yes, of course. The Microcycle platform, in essence, is a different, we'd like to think more powerful way of discovering new drugs, new hits and leads and drugs. And at the heart of that, there's really two separate components. It's what the library itself, what the components of that library are. And then the second part is how we screen. So the compound library is small cyclic peptides, and we call those microcycles. 6-membered is where we've started with. And these are interesting compounds in themselves because unlike bigger peptides, the kind of thing macrocycles that people talk about, which are large and floppy, microcycles are small and rigid and behave much more like small molecules than larger peptides or even proteins. So they fold predictably, which is really important too. They all adopt a kind of saddle shape, and that's because of the way the functional groups arrange around the ring. That's helpful for us. We can predict how functional microcycles are folding. But the one key difference, I think, between what we have as a microcycle library and, a small molecule library is it occupies a unique chemical space. So if we do chemical space analysis, we see a distinction between the kind of 3D chemical space that a small molecule library occupies and the 3D space that our microcycles occupy. And that helps us to address targets in a different way to a small molecule library. So different parts of the target in a different way. Perhaps we tend to find a lot of allosteric and cryptic sites on targets. So the microcycles are quite unique molecules, but I think the second component is really how we screen. So each library member we screen directly for biological function, and we do that inside a human cell, where the target of interest is presented, as I said earlier, in all of its disease-relevant conformations. And importantly, unlike conventional screening techniques where you'd use multi-well plates, 96 or 384 well plates, and a lot of robotics and a lot of plumbing to test each library member against your target, we can do all of this in pool. So we start off with a pool of cells, typically over 1 billion human cells, mammalian cells, and each one is expressing a single member of our library. And the cells that we use have been engineered in a way so that they can tell us that whatever's expressed in them, whether it has a desired biological activity. So it's a functional cell-based assay that's been set up. And this can be in HEK cells, for example, or we can use disease-relevant cells in the case of cancers. We can use cancers where the target is upregulated or plays a key role. And then, we let microcycles express. We're able to sort the cells where we have active microcycles from those that are inactive. We can do that using high throughput screening, for example, through cell sorters. We use very high capacity cell sorters. We can sort over 1 billion cells in a day now, and it's a very efficient way of finding biologically active microcycles. And because these are peptides that have been expressed in the cell, we don't just have the microcycles hits in each cell, we also have their genes. So we can use next-gen sequencing to understand which microcycles having which activity. We can make sub-libraries, new libraries, and screen again, 2 or 3 rounds of screening, each time enriching that little pool of cells for microcycles that have the greatest biological function. And then, very simple, we take those microcycles, we test them, put them in secondary assays, work out how they're working, how they're binding to their target, and then we can optimize them. We can optimize them in the same way that you'd optimize a small molecule, and that way we can find a way to, for example, a microcycle-based drug. That's one strand of what we're doing. But another strand of what we're doing is, because I said earlier, microcycles are small and rigid. That's quite unique in terms of cyclic peptides, and we can use that information to predict how the compound is binding to the target and how we can reproduce that in a small molecule. So, for example, in one of our programs, our HIF program, we've computationally worked out the position in space of the pharmacophore, the bit of the microcycle that's binding into the target, and we've been able to reproduce that in a non-peptide small molecule, and that has similar activity to the microcycle and which binds to the same part of the protein. So larger peptides, you make a large peptide, the peptide is the product. With us, we've got a choice whether we take a microcycle forward or we take a small molecule forward. Partly that will be driven by the target and the disease area and which one works better. But at least to some extent that option is there to us to carry on with a microcycle starting point or to move to a small molecule. And that approach, by screening in cells, doing that in the way we do it, particularly for intracellular targets, as far as I know, is unique. Even within this emerging space of cyclics, Merck have got their PCSK9 inhibitor, J&J have an IL-23 cyclic. Revolution Medicines are working on the RAS pathway almost entirely on cyclics. But the approach we're taking, we let biology do all the hard work for us. So yes, we use computational methods to help us at the end of the process, but we're letting biology do the work for us rather than having to create an artificial system. Another way in which that helps us is that some targets, it's very difficult to screen for them outside of a cell. We'll hopefully talk a bit more about our programs later, but one of our programs against a target called FOXA1, that's a really interesting target because if you take FOXA1 out of a cell, it misfolds. So you can't screen against it very easily or at all in an in vitro system. So you have to screen a library in a cell, and that positions us really nicely in the space for things like pioneer factors, transcription factors, protein-protein interactions, which is where we've had most of the success so far with the platform.
It seems like it's got very broad application potential. What conditions or diseases are you looking to address within that?
You're quite right, Jim. So it is almost completely disease area agnostic. For example, in the collaboration that we have with Merck, we're working on a neurology target. In our own programs, most of those are cancer programs, but we're starting to work on immune inflammatory conditions as well. So really the limiting factor for us is building the expertise in-house in those targets and disease areas. As a relatively young company, to work in too many disease areas is difficult. So we've tended to focus on cancer and inflammation, but there are almost limitless number of disease areas you can think about. And the way in which we can start to address those, is rather than sort of build all that expertise in various disease areas, is work with pharma on those targets. So where pharma wants to try and find modulators of targets in disease areas, they bring that disease area expertise and the target expertise, and we can bolt on our screening platform, and that way we can address the multitude of targets in various different disease indications that just off our own bat would be difficult to do.
Within your own work, then maybe you could tell me a bit about the assets that you have in your pipeline that you're working on and what their mode of action is.
Of course. We have 3 cancer programs currently, but they're all in preclinical phase of research and development. The one that's the most advanced is our HIF program. So HIF is hypoxia-inducible factor. And HIF is a transcription factor that's upregulated in very low oxygen conditions. So you can imagine in the core of a solid tumour where there's very little blood supply, very little oxygen, HIF is upregulated. And HIF then is a transcription factor which drives a whole range of other genes, for example, VEGF, and so you start to get blood vessel formation into the core of the tumour, and a whole load of other survival genes that allow those cells in the middle of tumours to survive. And it's an important protein. It was the subject of a Nobel Prize. It's one of those targets which has been of interest to pharma for a long time. And the sort of 2 flavors of HIF that we're interested in are HIF1 and HIF2. HIF1 and HIF2 drive different sets of genes. They have some overlap, but in general, they drive completely different sets of genes, and they're both important to the progression of tumours. But so far, only HIF2 inhibitors have been discovered and developed. So there's a HIF2 inhibitor at market. It's called belzutifan. It's been originally developed by a company called Peloton that were acquired by Merck. And Merck have developed it, and it's been now approved for about 5 years, and it's doing really well in a relatively niche disease called von Hippel-Lindau disease, a consequence of which is a number of different tumours, but the one that everyone knows is renal cell carcinoma. So it's approved as a monotherapy in those patients, and it's approved in combination for people with advanced renal cell carcinoma. But there have been no real successful attempts at inhibiting HIF1. And part of the reason for that is that there's a lot of similarities between HIF1 and HIF2. The big difference is that HIF2's got a nice druggable pocket in the middle of it, which Peloton were able to address with their compound, whereas HIF1, that pocket is very small. It's too small for conventional small molecule. So the approach that was taken was we originally screened against HIF2. Actually, the work was originally done by our academic in his lab, Ali Tavasoli. He screened against HIF2, back screened against HIF1, and found things that could inhibit both HIF1 and HIF2 roughly equally. And he identified through a second screen compounds that were just HIF1 selective. And we're looking at both. We're looking to see if HIF1 selectivity takes us in one direction in cancer, HIF1/2 would take us in a slightly different direction. But both have really strong potential in the treatment of particularly solid tumours, advanced solid tumours that evade conventional therapy. So the plan will be, once we've got a development candidate, to take that into the clinic. And depending on whether it's a HIF1 or HIF1/2 inhibitor will determine which patient population we go in. This will be a first-in-class, either a HIF1 or HIF1/2 inhibitor would be a first-in-class drug with enormous potential. Our second program targets an enzyme called ATIC. Now, ATIC is an interesting target. It's a well-known target, actually. It catalyzes the final two steps of purine biosynthesis. Now, purine is obviously very important to cancer cells because they're half of the bases of DNA. So you need a lot of new DNA in a rapidly growing tumour. And one of the ways in which the cell can make those purines de novo is using ATIC. Now, that's been known for a long time. And in fact, there have been a whole number of drugs that have been developed to inhibit ATIC, but they're all folate inhibitors. ATIC is a folate-dependent enzyme. And so, well-known drugs like methotrexate and pemetrexed, they've all seen some limited success in the treatment of cancer, but they don't really work particularly well. The reason for that is they're competing with a very large amount of folate in cells, so you need a lot of drug to overcome the native folate. And also, if you start putting a lot of drug into the cells, then you inhibit all folate-dependent pathways in cells, including in healthy cells. So getting that balance right between efficacy and side effects has been a real challenge for the antifolates. Now, what we're doing in a slightly different way is that instead of inhibiting the folate binding site in the enzyme, we're exploiting a particular characteristic of ATIC, which is that it's actually a homodimer. So there's two parts of the protein. When they come together, the enzyme becomes active, and what we have is a way of stopping that homodimerization. So it's stopping a protein-protein interaction. And if the protein can't homodimerize, it's not active. So this is a folate-independent mechanism by which we can inhibit the function of this really important enzyme. And it's particularly important in tumours that have a particular mutation and deletion in a protein called MTAP, because there's another way in which cells can get hold of purines, and that's through salvaging purines from its surroundings. And MTAP is responsible for that. But MTAP is quite often deleted or mutated in cells. It's co-located on the same gene as P53, and in a lot of P53 mutants, you also see MTAP mutation or deletion. So we're looking at, again, at quite a broad range of solid tumour patients that have that MTAP deletion where they're entirely dependent on de novo purine biosynthesis, and where we can block that biosynthesis in a way that could be done potentially with much fewer side effects and much more potently than you can achieve with a folate-dependent mechanism. That's a target that's been drugged before, but we can do it in a different way. Whereas HIF1 is a target that no one's been able to address before, and we've done it in a way that conventional methods can't. And then finally, FOXA1 on our cancer stream of compounds. FOXA1's a really interesting target. It's a breast and prostate cancer target. FOX plays a really key role in when you use hormone treatment in, say, breast cancer, things like tamoxifen or the aurora kinase inhibitors. There comes a point where the tumour becomes resistant to those. And it's not because the estrogen receptor has been mutated or has been eliminated. It's because the estrogen receptor has found a way of communicating with growth factors other than through hormonal stimulation. And the protein responsible for that reprogramming of the estrogen pathway is FOXA1. And it's been a long-held desirable target for pharma. And we believe that by screening in mammalian cells is the only way you can really crack this particular problem. And we've been doing that for the last couple of years. We've developed this kind of multi-billion cell screening approach. On the first screen we did of our large-scale screening, we screened 3 billion cells in a day. And now, after 3 rounds of screening, we're hopefully homing in on some functional inhibitors of FOX, which is going to be really exciting. If we can achieve that, it's a real feather in our cap for the company and for the technology. And as I said earlier, that will have application in both breast cancer and prostate cancer, where a similar mechanism takes place.
Does that mean that you'll be working just in drug discovery, or are you planning on doing clinical trials as well?
Yeah, for our own programs, obviously, we've got 3 programs. Following the Series A that we raised last year, is the plan to take one of those 3 programs forward into clinical development. We don't have the resources to do all 3 right now. And I have to say, right now, looking at the data coming from all 3 programs, it's an enviable choice as to which one we take forward because they all look interesting in their own right. But, with those 3 programs, we have opportunities to take one forward potentially partner one or two of the others at an early stage with pharma. As we move forward through into Series B, whenever that is, we can have more resources to take further programs forward and to initiate new screening programs. And it's a program I haven't mentioned. We have a RAS program, which, although we're not developing it ourselves, it's a really good example of how we can get the technology to work. So obviously, RAS has attracted a lot of attention over the last few years. Firstly, the G12C inhibitors, but now G12D and PAN inhibitors. It was a good workhorse system for us to use to develop the technology because it's a validated target, all the assays are available. And what surprised us, I think, when we did the screening was that we did find things that are inhibitors of the RAS-RAF interaction, and we've shown that quite convincingly now. But when we do the structural biology, what we found is a way of inhibiting the RAS that doesn't involve hitting the same part of the molecule that everybody else hits, which is called the switch 2 pocket. The switch 2 pocket in RAS is the pocket which converts non-activated to activated RAS. But we found a different part of the target which inhibits that protein-protein interaction, which is not the switch 2 pocket. And one of the consequences of that is that, we see that it can inhibit both activated and non-activated RAS in the same way. So that tells you a little bit about how the technology works, in that we're finding different things to the way you would find if you used a conventional approach. Even on targets where there's already considerable amount of small molecule work, we're able to do very different things. And that program, we're already looking for partners for. It's a very crowded area, but people are looking for interesting and novel things and things that behave differently. And that's all in that mixture of opportunities that we have.
So now you know what microcycles are. Very interesting technology and one more way we're standing up to cancer. Next, we have a conversation with Stefano Gulla, CEO at Amsterdam-headquartered Kling Biotherapeutics, and this gives us a chance to learn about B cells and their role in beating cancer. Hi Stefano, great to have you on the podcast, but before we get to B cells, could you fill us in a little bit on Kling Bio?
So Kling Bio, I like to think that we are working on solving what is the most pressing problem in biotech, which is the validation and the discovery of new targets. I think you know by reading news and being informed about the biotech field that there's a huge focus on about 20 well-known targets. There's hundreds of programs that are focusing on the same biology. And what that does is produce an environment in which there is very little interest and knowledge about new potential therapeutics that can come up and really have a transformational change for patients. What we're doing here is we're using the human immune system as our discovery tool. And we can do that because we have proprietary technology that allows us to look very deeply into what the B cells, which are really drug-making factories within the human immune system, what they're recognizing and what they're finding that is unique about infectious disease or cancer. We use that information to discover new antigens, which then become validated targets. And we go after these targets with all the tools that modern biopharmaceutical discovery allows us to do. So therapeutic antibodies, ADCs, bispecifics, and so on. So by doing that, we have not only discovered and validated a number of new targets in the oncology field and in the infectious disease field, but we've created a pipeline of these agents that are now moving forward preclinically towards the clinic.
I think within the field, most people would have heard of T cells, but could you give me a bit more information on B cells and what they are?
T cells recognize antigens as form of peptides, which are displayed by cells. B cells produce antibodies, and antibodies are proteins that are able to recognize more complex antigens, so proteins that are folded differently. The real role of the immune system is very simple. It's trying to determine what is foreign in your body. So it's to distinguish self versus non-self. And this is where antibodies really come in. They're able to recognize very minute differences and be very specific about what makes an antigen non-self. This is obviously very important when it comes to fighting foreign antigens such as viruses or bacteria. And that's where antibodies are very well known for, to be able to neutralize viruses and therefore allowing us to live a healthy lifestyle. What we've learned over the past 15 years is that the immune system is also capable of recognizing antigens that are expressed by aberrant cells like tumors. And what we're doing here at Kling is leveraging the B cell response, so the production of these antibodies by the human immune system, to identify which antigens of cancer cells are unique and can be leveraged to develop new therapeutics. What's really, again, really interesting about B cells is the fact that not only they're able to recognize these antigens, but they produce the antibodies as really ready-made drugs. What allows us to leverage this power of the B cells is the fact that our technology is able to take this information out of the in vivo system, so out of the body, and put it in the lab in the form of immortalized libraries. So we can immortalize about 80% of a sample of B cells that we derive either from peripheral blood or even directly from tissues. And these libraries can then be assayed via high throughput means to identify what they are expressing and what antigens they're recognizing. So we've spent a lot of time over the past 2 years to hone in our skills and our throughput technologies in order to go from immortalized libraries to validated antigens and antibodies as quickly as possible. And we can do that within a few weeks, go from a primary B cell sample to a hit, which is an antibody that recognizes something unique about a particular cancer antigen, or even in the context of an infectious disease, a neutralizing antibody.
And as far as your programs are concerned, could you tell me something about Kling Select?
Sure. We have some nomenclature here, Skillington. We call Kling Select is the process of taking primary B cells from a patient or even healthy volunteers, immortalizing those B cells, so therefore creating these living libraries of B cells that can be expanded, frozen, and re-assayed many times. And then in addition to that, what we add to this, what we call this Kling Select module, is the fact that we can also then screen the B cells via a function-first approach. What that means is that we're not simply looking for antibodies that bind specifically to a particular target or cell line, but we're also looking deeply into the function of those antibodies. So we can, for example, immediately identify neutralizing antibodies against viruses like COVID, influenza, for example. We have some of those programs. But also we can find antibodies that have a specific function with respect to our cancer cell lines, whether they have direct cell-killing abilities or whether they have internalizing abilities that we can then leverage as antibody-drug conjugates to deliver toxic payloads. So what we really call Kling Select is this process of taking primary B cells and at the end of it, having functional antibodies that can be leveraged as therapeutics.
And what's then Kling Evolve?
Kling Evolve is really unique. So I come from a antibody engineering background. Well, one thing that really struck me about our technology is the fact that once we immortalize the B cells, they're still fully functional, and they do whatever B cells are really excellent at doing, which is to adapt to an antigen to create even greater selectivity and potency. Kling Evolve refers to our ability to take these interesting B cell clones that we identify with Kling Select and then further improve them or optimize them via a process called somatic hypermutation. So we can take the clonal B cells from Kling Select, allow them to grow under certain conditions and produce additional genetic diversity. And then we can rescreen these new libraries for higher affinity, for example, or higher potency or better specificity. So essentially, we are evolving the B cells that we have identified in order to be even better therapeutic targets. And we've done this for a number of targets, but very recently we published a paper in Frontiers Immunology where we show this approach is able to very rapidly evolve higher potency antibodies against new strains of SARS-CoV-2. So what that means is that even though the virus is constantly changing, we can use our ex vivo libraries via Kling Evolve to adapt to the new variants as they come up. Therefore, we can co-evolve the antibody with the virus and have a very quick response to emerging variants that could be of concern.
So where are you at currently on your journey to treat cancer and other conditions?
Our platform is naturally suited to infectious disease, and we are actively pursuing a number of opportunities there, including COVID or even flu-targeted antibodies. But we're also very focused on our oncology pipeline. We have prioritized 3 of our initial targets and antibodies. And one of them in particular is very interesting, is an antibody that binds a glycosylated form on a protein called CD43. We found this antibody by screening patients that had recovered from AML after allogeneic stem cell transplant. So this antibody recognizes a glycoform on a target that was previously undescribed. So it's a completely new antigen that is very specific for AML. And what we have found is that the antibody is also able to internalize very effectively on AML blasts. So we're developing this antibody as an antibody drug conjugate. We have a lot of hopes as this is our lead programs in oncology. We are now starting our preclinical development there. We have other targets as well for other hematological conditions, as well as a target for solid tumors, which we're also advancing at earlier stages of discovery.
Are you able to tell me anything about the partnership with Sanofi?
Yeah, it's a really important partnership for us. First of all, it validates the power of the technology and how external parties such as Sanofi, which is a world-class pharmaceuticals, are able to see what it can bring to their own pipeline. It's focused on discovering new antibodies and new epitopes that could be potentially then be leveraged as vaccines. We're not disclosing the target, but this is a very common respiratory illness that we're going after here. It's a multi-year collaboration. The collaboration here involves our Kling Select technology applied to a number of patient samples to extract information about what antigens the antibodies are recognizing. And then Sanofi is going to take on that information and potentially develop new vaccines or potentially new therapeutics out of that.
As this is going to be a part of a special about cancer, I wonder if you could tell me what you think is going on in terms of the fight against cancer, where we're at, and your hopes and goals in that respect, both generally and from your company's perspective.
I think we are at a stage now where we have been able, as a pharmaceutical industry, to come up with a number of different opportunities for how we address cancer. So we have I call this as basically tools in our toolbox. So we have, for example, CAR-Ts, ADCs, therapeutic antibodies, bispecifics. So we have all these tools, and I think there's a lot of interest now in applying these tools and see how far we can take them with respect to new targets. I do feel that there is, as I was mentioning earlier, a little bit of an issue with how the industry is prioritizing new targets when it comes to the next steps. So there's definitely a conservative approach now to going after well-validated targets that can then be made better by application of these new tools. We see that there is a bigger opportunity by going after what we think are new targets. We are looking for transformative outcomes in patients. And I think that that's where the next step of the oncology field needs to be, is now taking the tool sets that we have validated over the past 15, 20 years and applying them to new biology and seeing how far we can actually take those from a point of view of true recovery and outcome for patients. So I'm excited about that. I'm excited about the role that we can play as Kling to bring forth some of these new targets. And also excited about potential collaborations that we are having with partners about applications of our discoveries with new therapeutic modalities that can even further increase their potency. So we are in a time of change, but I do think the future is positive, and with a little bit more courage on the side of investors, I think we'll be able to make transformative leaps in the next few years.
And I guess it's difficult because there are so many different types of cancers, and some of them are a lot easier to take on than others. There are some that are really difficult. So there's kind of the two extremes.
There are definitely indications in cancer where we had great successes. For example, B-cell leukemias, we have been able to transform that into basically a curable cancer with cure rates in the 80s to 90%. But there are areas where we haven't advanced as much. And this is also where we're focusing our search. So our lead target is going after AML, which is a myeloid leukemia, which is has a high level of mortality, and also where there hasn't been as much of a advancement in how we treat them. So we're looking for new targets in areas where we see a high level of unmet medical need. I think that's where we can make our biggest impact.
Do you think that that's because the more difficult ones people shy away from, or is it just that it's really difficult to target and people are trying?
I think there's definitely not for lack of trying. We have tools at our disposal, like genomics, proteomics, that have helped us in some cases, but in other cases, they've come up short of really pointing us in the right direction. I do think that our approach is different, and it can really add to the sort of target space that we can access in these diseases. So that's why we think that going after these highly refractory diseases in which we haven't had success with conventional approaches for finding new targets, that's where we can have our biggest impact. The unique things here is that the immune system can identify differences that would be completely overlooked by other methodologies. Again, going back to our lead asset, is a glycoform, which would be completely unrecognized by a genomic screen or even a proteomic screen, because the protein is still there, but it just has a different sugar displayed on the surface. The great thing about the immune system is that it's able to find these very minute differences between a disease state and a non-disease state, and we can leverage that with our technology. So we think that we have something really unique here in the way that we can look more deeply into the immune response and derive insights that would be otherwise impossible to find with other means.
More great things happening there in Amsterdam with Kling Bio. You get the feeling there are going to be a whole lot of breakthroughs in the coming years, and it can't come soon enough. So the next company we're going to hear from is Poolbeg Pharma, another company with an awful lot going on. One of the potential issues with immunotherapy is the possibility of cytokine release syndrome, which can also affect those recovering from COVID and other conditions. Poolbeg is addressing this issue, and to tell us all about it and the company's other work is CEO Jeremy Skillington. It's a pleasure to talk to you again, Jeremy, and maybe we could start with a bit of background on the origins of the company.
Yeah, happy to do that. Yeah, so Poolbeg Pharma, we are listed. We are headquartered in London, listed on the London AIM market. We spun out of another AIM-listed company, hVivo, back in 2021. And interestingly, hVivo they run human viral challenge studies. So, quite, in the middle of the pandemic, here was, quite a successful viral testing company. And they also had a biotech arm that at the time was like they were running two strategies at once. So we thought it was more of a clever option to spin Poolbeg out of hVivo and run it as an independent biotech company. We raised separate financing. We brought in a separate management team. So we were going since 2021. And interestingly, initially we were focused in on severe influenza. And we had a treatment for severe influenza, and it's calming the cytokine storm that's associated with severe influenza. And as we were pushing through our development, we ran a clinical trial to test the hypothesis that our lead program, POB-001, could indeed inhibit an inflammatory response in humans. And as we were progressing down, we got very good clinical data. But then we looked around and we saw that severe influenza is an unmet need, but an even greater unmet need is a condition called cytokine release syndrome associated with cancer immunotherapies. And I'm sure, Jim, we will go into great depth on that. But we then kind of pivoted, you could say, the company to look in that direction. So we've been pulling together clinical trial activities to begin a phase 2b trial to prevent cytokine release syndrome in patients that receive these cancer immunotherapies. So as I said, we've been moving very quickly, on that front. We also have a couple of other programs in our pipeline. We have through a collaboration, an oral formulation of GLP-1. So again, the world is getting very excited with the potential for GLP-1s. Particularly in, initially it was diabetes, but then more recently in obesity conditions and corollaries around cardiovascular disease and renal disease as an example. So again, looking to push that forward to get some clinical data in that setting. And then, thirdly, as a company, we did some AI analysis of data that hVIVO provided us in influenza and RSV, and we've identified some very interesting drugs and targets in that space and looking for partnerships there. So we're a small company, but it's a very ambitious, publicly listed, and got some clinical programs in the very near future that we're looking forward to executing on and seeing the data, of course.
Could you maybe go into a little bit more detail on cytokine release syndrome and what it is and what's currently the status of the field in that, I guess?
Yeah, I will kind of take a step back and see that this is a relatively kind of new phenomenon or a new and growing phenomenon. I think the condition has been more recently understood in the context of these cancer immunotherapies. So what I call these T-cell engaging therapies. And this is, for example, CAR Ts are the initial example. But more recently, there's bispecific T-cell engaging antibodies. And these are very clever therapeutics in the cancer setting, because what they do is that they bring a cancer cell essentially in close proximity to your immune system, in particular T cells. And in that close proximity, the T cells recognize cancer cells, the fact that they shouldn't be there, and then the T cell actively kills the cancer cells. And very clever technology, but the efficacy has been terrific. You look at conditions, particularly in the blood cancers like multiple myeloma, you've seen terrific efficacy in that particular setting. People are very reluctant to talk about cures in cancer for obvious reasons, but there is acceptance that these T-cell engagers do deliver cures to certain patients. But what's the issue then is that they have a side effect, because as with everything, when you're manipulating the immune system, you're always keeping an eye on how the immune system ultimately responds. Yes, positively, you do see that reduction in tumor burden, but you also have this issue around cytokine release syndrome or cytokine storm, and that happens in about 70%, roughly 70% of the patients. So it's a known issue, it's a known side effect. And what happens then, as I say, the patient's immune system kind of gets a little out of control. So it's you're triggering the immune system to go after tumors, but sometimes then it goes kind of awry. Let's say, I like to say, like the plane has run off the runway, in that it's a little bit out of control. So you need to bring that in control. And it can be kind of significant. As I say, it starts with kind of low blood pressure. The patients are in a very uncomfortable position. It can escalate to kind of significant issues where they need respiratory interventions, for example. There have been examples where patients have died as a result of these cytokine release syndrome issues. And as I said earlier on, with these T-cell engagers, now on the market, many more coming through, it's a known issue. So patients receive these therapies in a hospital in a dedicated cancer setting, knowing that, as I said, 70% or so will develop CRS. So the docs and nurses are on hand to, when they see the evidence that CRS is developing, to intervene, as it were. So, as I say, they have to be in a hospital for this setting. But what we believe we have ourselves with POLB 001 is a preventative therapy. We can give the drug in advance of the cancer immunotherapy to prevent CRS beginning in the first place. So that's our hypothesis. We've got very compelling data, both preclinically and clinically, that suggests that this is a reasonable hypothesis, but obviously we want to test this in a clinical setting. That'll happen later this year. So you can imagine with all of these T-cell engagers, if we can remove CRS, that removes a lot of issues, a lot of, the kind of healthcare burdens. The patients can actually, stay at home, as it were, after receiving the therapy. And, prevention is always better than cure. So we see great potential for our drug in that setting.
Would it also be applicable to things like COVID, where CRS is an issue there too?
Yes, I mean, this is where we started, as I mentioned, around severe influenza. And yes, there are certain kind of corollaries. It is a case of the cytokine storms, as people remember from the pandemic. That, again, that's your immune system is out of control. It's triggered by the virus. A lot of times the virus has been eliminated, but the immune system is still rumbling on. It's still, as I say, in this kind of heightened sensitivity, heightened inflammatory mode. And yes, we believe we have the potential to impact or inhibit the cytokine storm that has occurred. But when we looked, from a company standpoint, a strategic standpoint, we looked at, the markets, and we saw the unmet needs, and we saw, as I mentioned, the explosion in the use of these T-cell engagers and the high incidence of the cytokines, release syndrome that began. We thought this was a better way to test our patients. And, we did approach many pharma companies who were in that kind of T-cell engaging space about our hypothesis and got some really strong interest. So we're, as I say, running a clinical trial later this year, beginning later this year, where we're going to give our drug in combination, you could say, with an approved T-cell engaging bispecific antibody to, again, measure and to determine the rate of CRS and if we're able to reduce that rate of CRS. So very excited again to move that forward. But you're right, we can see this kind of applicability, and this happens a lot in the industry. You get approval in one indication and then you can kind of branch out into others. But the same hypothesis, we believe, can have the same effect. We're hitting a target called P38 MAP kinase, which is well known to be a master regulator of the immune and inflammatory response. So again, that can overlap to other diseases and other indications.
Is it something that other companies are taking interest in? I talked to a company a few weeks ago that was looking into CRS. So is it common?
There are companies looking. It started off in a more of a repurposing situation, like what approved drugs can we put into this new, situation, this new indication, and that's where they came up with again the current treatment is a drug called tocilizumab. It's an antibody from Roche that initially was approved. It was indicated for rheumatoid arthritis. It hits a target called interleukin 6. So they know that interleukin 6 levels are elevated in CRS. So they use this and, have seen some success. But I'd say it's an expensive drug. So it's kind of given, again, in that reactive manner. You have to wait for the patients to develop CRS and then you give them tocilizumab. There have been clinical trials where they've given tocilizumab in advance, similarly in a prophylactic setting. Now, number one, when we talk to clinicians, recently at a conference in Milan, the European Hematology Association conference, that's nice hypothesis, but it would be very expensive for the healthcare systems to be giving all patients, an antibody in advance of getting their CAR T therapy, for example. But they've also shown that you're not reducing levels of what is called grade 2 CRS. So there's advantages. So everyone's looking for a better solution. And our drug in particular is a small molecule. It's orally delivered, so the patients can take this at home. So there's that convenience factor. But again, as I say, the prevention aspect is really what's getting people excited. And we did run a clinical trial, an LPS challenge study, where we triggered a patient's immune response using a bacterial membrane fragment. And what we've seen when we gave our drug in advance, that we were suppressing that immune inflammatory response. And I will say we looked across multiple cytokines, including TNF alpha, but interleukin 6 as well. So we see a decrease in a multitude of these cytokines when we inhibit or target P38 MAP kinase. So we see we can have broader application. Let's say we're not just hitting one cytokine, we're hitting many. So again, as I said, the point is, the team is working hard to get all the pieces in place to start this clinical trial later this year. And I'm so excited to see what the data looks like next year.
Could you tell me a little bit more about your lead candidate and how it works?
Yeah, as I said, POLB001, small molecule P38 MAP kinase inhibitor. This was a target of significant interest to pharma companies maybe 15, 20 years ago, as pharma is wanted. If they get an exciting target, they all roll in behind, get their chemists developing therapeutics inhibitors. So they're looking at the time these blockbuster indications like rheumatoid arthritis, and many companies ran several clinical trials looking at this. But what they discovered with P38 MAP kinase, it was a very good target to inhibit this kind of immune inflammatory response, but it couldn't be applied in a chronic setting. And as you can imagine, if you're diagnosed with rheumatoid arthritis, in your 50s, you're going to be on therapy essentially the rest of your life. So they couldn't give this drug long term because you had some liver enzyme elevations. So what we discovered, what we kind of applied, is that it's a perfect drug, a perfect target for these acute inflammatory conditions where you don't have to give the drug long term. And that fits perfectly into the mechanism of cytokine release syndrome, where you're preventing it, you're giving the drug in advance, you're suppressing the immune response, and then you take them off drug, let's say after 8 days, 10 days, 2 weeks. They're only on drug for that short period of time. So that's, we see great viability, a fantastic target. But now I think we found the optimal indication, since these new cancer immunotherapies have come through. So we see this, we've got terrific data in humans, both safety aspects and as I mentioned in that LPS challenge study. So very happy with the data we have in humans, and even preclinical preclinically, preventing CRS from developing in animal models. So we have a nice cohort of data that, as I say, gives us strong belief that when we put this drug in the clinic later this year in actual multiple myeloma patients, that we will also see inhibition of CRS. So yeah, there's other competitors out there, but I think we found a nice application for p38 MAP kinase inhibitors.
And what does the clinical trial pathway look like? I know you're mentioning that later this year, but how's that fit in?
Yeah, we've got a plan. We're working with some principal investigators in the UK. We're looking at an open label study. So this will be roughly 30 patients who have been diagnosed with multiple myeloma, and they're about to receive T-cell engagers. So as I mentioned, we will give our drug in advance, keeping the immune system in check, and they will receive their bispecific T-cell engagers. The analysis is pretty rapid because, again, we're not looking at that kind of tumor response. This is an approved T-cell engaging antibody, so we understand the historical data, but we're looking at that immune inflammatory response. So it's a relatively rapid clinical trial. As I say, we're just looking at the immune response in the first, let's say, 30 days. And then, as I say, seeing that we're keeping this cytokine storm, so CRS, under control. And because it's open label, we're able to enroll a patient, look at their data very rapidly. So there'll be plenty of opportunities for interim analysis. And again, as I mentioned, in the standard of care, as it were, you're expecting roughly 70% of patients to develop CRS. So we're going to be looking closely at the percentage of patients that develop, if indeed they do develop CRS. So we'll be able to get very quick and rapid readouts. So we'll have interim data in the first half of 2026, and then top line data in the second half of 2026. So very rapid clinical trial. And I think that's, from a Poolbeg standpoint, we're very keen to partner our program with big pharma companies. As mentioned, from a business development effort standpoint, we've talked to all of them. They're very intrigued to see the data, certainly the data in patients that we'll have next year. And from a Poolbeg standpoint, then that we believe that will trigger partnership opportunities where they will then kind of progress that further into the clinic, phase 3s and ultimately onto the market. So we think that we can solve a problem that they have, and believe that if it's successful in this clinical trial with one bispecific, you can apply it to all other bispecifics and indeed CAR Ts when it comes to those that, as I say, trigger that CRS. But prevention here is the key aspect, and I think that's a key differentiator from a Poolbeg standpoint.
Yeah, you mentioned the fact that it's prevention, i.e. giving it before treatment. What happens if you were to, like in COVID or influenza, where you get CRS? Does the stage at which you give the drug make a difference in cases like that?
Yeah, that's a really, really good point, Jim. You're right. When we started from a Poolbeg strategy standpoint, as you mentioned, severe influenza, where we began. And again, when we spun out of hVIVO, the team there had done some terrific work looking at severe influenza and normal influenza response as well, and seeing that p38 MAP kinase was a key driver of that transition from mild to moderate influenza to severe. So that's why they went after it. We believe that then when you inhibit that, you're going to calm the immune system down. As you say, let's say the horse is already bolting with regard to the cytokine storm has begun. So we believe if you block p38, you can kind of reduce that effect, because it is in that kind of vicious cycle. There's a continuum of the immune inflammatory response. Like cytokines are released or trigger migration of recruitment of immune cells, who then release more cytokines, who trigger recruitment, and it's a vicious cycle. So we think the p38 MAP kinase can block that cycle. So that's on the one setting. But when we looked at as I mentioned, the LPS clinical data we generated, prevention is obviously more of an attractive, but more applicable to these CAR T cells and bispecific antibodies, because you know when the patients are going to receive these antibodies, so you can kind of give our drug in advance. So it's a very controlled setting, obviously, and what we expect again from preclinical data is that it doesn't, and this is very important, it doesn't impact the T cell activity of these therapeutics, but it does keep the immune system under control. And again, importantly, one piece of data that we saw from our clinical trials, it doesn't give 100% inhibition of your cytokines, because you need some intact immune system around just in case you get an infection, for example. So what we're seeing is that we believe we maintain that intact immune system, but we don't allow it to get out of control, when triggered by these bispecific or CAR T cell therapies. And again, that's really important for the cancer patients, because obviously they're in a difficult situation themselves. The last thing they want is an infection, following treatments. But so we believe, as I say, we're kind of threading the needle in all these important aspects of what a successful drug, a successful prevention of CRS drug looks like.
And do you have other things in your pipeline other than tackling CRS? Not that that's not big enough on its own.
It's a good point. That is our lead asset, lead program. We do have a very interesting collaboration with some partners who have an encapsulation technology. So what we're looking at here is encapsulating the GLP-1 peptides. So again, GLP-1 have been growing dramatically as a drug class over the last several years, but they were, for the majority, all but one, they're given by injection. So these are kind of daily injections or weekly injections to kind of keep your GLP agonists, and they've seen tremendous benefits in, as I mentioned, diabetes, in weight loss, obesity, and other associated benefits. But we've got this encapsulation technology where you can give the drug orally. It protects the GLP-1 peptide from the stomach acids and enzymes. It gets into the small intestine. To change of pH, you see it then kind of is released into the small intestine, which coincidentally and happily is the site of action of the GLP-1 receptors are present in the small intestine. So we've again got a clinical trial planned, running this with Professor Carel Leroux, who's very globally recognized in the metabolic disease space. And so we're going through the kind of manufacturing aspects right now with a clinical trial to be run in Northern Ireland. And again, similar to POV 001, these are very short clinical trials, so we will have our top-line data in the first half of next year. As a small, ambitious biotech company, getting that clinical data as quickly as possible is very important to us, and obviously important to our collaborators, to potential partnerships and investors. So we're very keen to get that data as quickly as possible. And if we do see, Jim, if we do see that we're seeing biological activity of the GLP-1 receptor agonist peptides, that kind of gives us convincing that this is a viable drug, that they can be given orally, and obviously the market for these GLP-1s is quite large and growing. I think people are very excited about the potential. It's without question the market is competitive. But we, again, from a business development standpoint, have talked to all of the key leaders in the GLP-1 space, telling them our story, telling them about our technology, what we're doing, and again, very keen to see the data that we're generating. And everyone is trying to kind of elbow into this, what's essentially $150 billion US dollar market by 2031. So, they're looking for these edges, looking for advantages. And as I say, if you can move away from injections, many patients would be more than happy to take the drugs. There is kind of a high kind of dropout rate of people with needle phobia, for example.
Is there anything else that you wanted to mention?
We've got a terrific team here at Poolbeg. They do a lot of work in the background. Very excited to work with those. And I'd say just our investors, again, have really happy to engage with them, keep them up to date on what we're doing, and very excited now for this next stage of Poolbeg and this kind of clinical data as it rolls out, and sharing that with investors and with the market, etc. So exciting time for the company, and looking forward to seeing pushing everything forward.
We've talked about CRS a couple of times on the podcast recently, so it seems like there are going to be options for treating that in the near future. The last thing that people with a terrible disease like cancer need is side effects. Anyway, that's it for another podcast. Thanks for joining us as we stand up to the various forms of cancer, and the industry does its best to make cancer a manageable illness or something that can be cured. Let's hope that it's sooner rather than later. So until next time, wherever in the world you are, I hope you have a great week. Thanks for listening, and I hope you'll join us again next week for another Base to Base Biotech podcast. The Base to Base Biotech podcast is an Air Coastal Media Limited production.